Okay, thank you. So, I am Ingrida, simple Ingrida, and then my research interest is invertebrates, some special groups of invertebrates like bryozoa, tardigrades, and leeches. And so today I would like to present some general information about invertebrates about aquatic invertebrates in the rivers and the streams, about their functions in water ecosystems, and how you can collect, and why you need to do so, and about the importance. Water quality is very important for us and for various organisms which live in the water, of course. Scientists which want to evaluate water quality usually apply it to methods, so chemical and biological. And it is good because both of these methods have some negative and positive features. So if we want to evaluate water quality using chemical methods, of course, we will obtain rapid and enough exact answers about water composition. It is good, it is positive feature. But negative feature that these results reflect water composition at the moment of sampling, mainly. And it is negative feature of chemical investigation. Another way, or another method, biological monitoring or evaluation of water based on living organisms, also have some negative and positive features. And then different from chemical research, these living organisms provide for us information about water quality over some period of time, different from chemical investigation. And therefore, this method is quite useful. But also, this method have some negative features because you need to have enough extensive knowledge about invertebrates taxonomy. You need to know how to identify them. And also some invertebrates can be affected by seasons, for example, or some changing velocities, some depend on some special life cycles. So therefore, of course, it's very useful to combine this both methods, chemical and biological. And based on both these methods, we can evaluate water quality. Rivers and streams are dynamic systems and home of various organisms. Of course, you can find in the water, and possible you will try to find some invertebrates today. You will meet and you will find various invertebrates. I think that mostly your organisms which you will find will be invertebrates. So it is rivers and streams homes as also of various organisms. I would like to ask you, because today I would like to talk about invertebrates, macroinvertebrates. So I would like to ask you, please explain or define how you understand what is water macroinvertebrates, or how you can define what is, or what are, sorry, water macroinvertebrates, how you can define. These invertebrates, which you can see and observe by naked eye without microscope, is macroinvertebrates. And what does mean invertebrate? All organisms which have not the backbone, so invertebrates, so macroinvertebrates. And what does mean benthic macroinvertebrates? So it is an organism, or there are invertebrates which live on the bottom. So macroinvertebrates, small invertebrates living on the bottom, which you can find and to see and observe by naked eye without microscope. And the second question for you how do you think, what is the importance of these invertebrates in the water ecosystem?
Of the quality of water, for example, so specific species live in specific conditions, for example.
Yeah, but this importance possible for us, yes, because we based on invertebrates can orientate about quality of water, but possible I will ask by another way, what is function of invertebrates, or why they are important in the water ecosystem, what function they perform in the water ecosystem?
They are food chain, they are food for fishes.
Yeah, so they are very important like primary food source for various fishes and for various other organisms. And they exist, of course, like so special link, which helps to connect algae, plants, so producers can connect with the predators, for example, because this material from the plants, from producers, travel through the invertebrates like food source to the other levels or in the food net. So, because due to these functions, they are quite important in the water ecosystems, and here you can see various groups which represent macroinvertebrates, benthic macroinvertebrates. Of course, numerous number of various species hides under this one term. For example, if you can see here term leeches, which represent oligoheads. So it does not mean that finding and identification of leeches will be very easy. Under this one word hides about 10, for example, or 12 species, and of course you need to know how to recognize them. So if we're looking at this point of using macroinvertebrates like indicators, it is, we can say that it is a negative feature. Because some groups, of course, we can identify quite easy. Main taxon levels, we can identify quite easy. We can say that there are oligoheads, there are larvae of insects, there are crustaceans. But some of them we can identify quite difficult, and for identification of so groups we need to have enough extensive knowledge. But experience say that after some experiments, or some trying to collect and identify, people quite easy and quite rapidly obtain some features which allow them distinguish between main groups of invertebrates. Okay, and here you can see that insects, and only a few examples of insects, they consist the main part of macroinvertebrates, which you can find in the water ecosystems. Because, of course, insects are dominated class, and they have various development, and some parts of insect develops in the water. And sometimes you have not possibilities to see the adults insect, but you can quite easy to find these development stages or larva stages of so insects in the water, and then you can recognize and tell about these insects. Water, we said that invertebrates can tell us about quality of water, so we can use them like indicators. And possible, you know, that exist various kinds of pollution, so like organic nutrient pollution, toxic material pollution, some heavy metals pollution. And invertebrates or macroinvertebrates are usually used like indicators for evaluation of bioorganic nutrient pollution. For this type of pollution. And again, I will ask you, possibly you know which advantage features or positive features have macroinvertebrates, which allow for us use them for indicators. Possible you can list which features or which characteristics have invertebrates, and because this characteristic, positive characteristic, we can use them like indicators. We must study organisms in like indicators in the rivers and streams, in the flowing water, algae, macroinvertebrates, and fishes. So both, all these groups react to the various stressors in the water, and they choose like indicators because they provide good response, clearly visible response to various pollutants, and invertebrates too. So I come back to my question. Very nice, very good. One positive feature of macroinvertebrates, because they are abundant. Because they are abundant, it is quite important, because we can go to the river, to the stream, and then we can expect where we will find these invertebrates. We saw in the table that various groups of invertebrates live in the water. So they quite diverse. And then especially diverse insect group. And depending on diversity, of course, we can choose and then evaluate water quality. So it is quite easy if we have wide various groups of invertebrates. And
that's difficult to identify. I mean, maybe not to species, but at least to the.
Yeah. So he is so doubtful about this feature, but yes, some groups of macroinvertebrates we can identify and recognize quite easy, as I said, at the main taxonomic levels. So like phylum, orders, sometimes family. Of course, exist some groups we can identify quite difficult. Identification is quite difficult. Yeah, and
short life cycle.
Not very exactly. Some of them spend their life, the majority part of life in the water. It is quite long period. For example, exist and some insects which live like adults in the terrestrial environment very shortly, one day or only a few hours. But whole life of these insects take place in the water environment. So majority of them spend more time in the water, and it is good feature like indicators, because if we constantly sit in the water, if we constantly sit in the same place, so they can absorb and concentrate some various materials from the water, and possible those materials will affect them. What about movement of invertebrates?
They are not that. They are not moving long distances.
Yeah. So it is some additional good feature using invertebrates like indicators, because they more or less sit at the same place. They can move, of course, there exist some invertebrates which constantly spend the time on the same place, or like sessile or settled invertebrates. But some of them, of course, they can move. We can travel or swim, but we'll do so in very short distance. So we can be sure that we can go in the same place, and then we can find the same or similar invertebrates in the same place. So it is a positive feature. Okay, here these features. Listed these features which allow choose macroinvertebrates like good indicators. And some of them were already said. So very common, as well already said. It is good feature. So taxa with different pollution tolerance. As invertebrates are quite diverse, And of course, we can, between them, we can find the groups which are very sensitive for various changing of environment. And some of them can quite easily adapt and survive in hard and changing water conditions. So, and between majority of taxa, we can find various groups which was exhibit different sensitivity to various stressors. Continuous monitoring, so as we said, they exhibit so limited locomotion, and at the same time we can provide with continuous monitoring on these invertebrates. On mobile, the same, inexpensive equipment. Inexpensive equipment is not very true for biological monitoring or for studying invertebrates in the water. Because, of course, for chemical studying of the water, we need to have various chemicals, and we need to have some special equipment. And this equipment, of course, is expensive. If we'll evaluate water based on macroinvertebrates, one part is enough cheap, because using enough cheap equipment, we can collect invertebrates from the water. So for collecting, for sampling invertebrates from the water, of course, we can use enough simple devices like nets, like buckets, like spoons or trays. So this equipment we can find in the kitchen. So it's not expensive. Collect them. But if we need to perform analysis of invertebrates collected and relate this analysis, connect this analysis with water content, water composition, water condition, so we need to collect not only biotic factors like invertebrates, but also we need to collect information about abiotic factors in the water. So as temperature, oxygen level, nutrient, organic, salt in the water, all pH, we need to determine these abiotic factors. And for determining these factors in the water, of course, we need to have some special equipment, which is not cheap. So it's not inexpensive equipment, it is not very true. So for collecting, this equipment is quite simple. But for more deeply analysis, we also need to have some enough expensive equipment. About identification, we already said, and the last tip, so no need chemicals. Of course, for studying these invertebrates, we not use chemicals except ethanol or few drops of formalin. It is true. So here you can see a few main habitats in which you can find benthic macroinvertebrates. I will talk only about benthic macroinvertebrates. So main types of substrate or habitats which you can find in the flowing systems. And the first is stubble substrate or hard substrate, which is made of mainly of stones, which covers the bottom, and this habitat is very rich and suitable for invertebrates. Very interesting habitat. And then, for example, I very like to study and to seek various invertebrates in soil habitat, because each stone can contain on the bottom of stone you can find various larvae, various mollusks, various organisms. And each stone can be like interesting discovery for scientists, for you. And the second, it is snaps. Snaps, various branches or wood debris, quite long time submerged underwater. And then so snaps, which quite long time are submerged, they contain various invertebrates which like to attach to these branches and then spend time, majority of time under this snacks. Vegetated banks, so various macrophytes and plants which grow in the water also contains varied invertebrates. Some of them we can find on the bottom part of stream. For example, various leeches, mollusks, some larvas you can find in submerged part of plants. And submerged macrophytes, they represented mainly various algae. So some invertebrates you can find and on these macrophytes, but it is how diverse invertebrates will be. If you will try to collect more macrophytes, it is dependent on species of various these macrophytes, because some of them not exhibit a great number of invertebrates. So simple invertebrates don't like to sit on some special kinds of macrophytes. And the last type habitat, it is streams, for example, which have sandy bottom and sandy or gravelly bottom, poor habitat from the point of invertebrates, because between these small grains of sand, invertebrates don't like to live, and usually so streams they have not or have quite small quantity of organic material, so simple invertebrates cannot find the suitable food for them in salt bottoms. Okay, and here you can see, like example, if you will try to investigate flowing water like streams or river, you need to choose habitats, or how many, you need to know how many habitats you need to study and which habitats you need to collect. So here, like example, you can see if you will try to study some stream, you need to choose some segment of stream which exhibit various habitats. Of course, will be good that this segment, chosen segment, represent the major characteristic of the whole stream. But also, if you will choose a segment with various habitats, so you will collect various invertebrates, and you can provide some special characteristic about the full stream using some special segment. Usually, it is recommended to choose the stream segment which in the distance 100 meters long. 100 meters long, and if the segment in this segment have diverse habitats, you need to choose the samples, some special number of samples in these special habitats. For example, here, if you have a stream which contains multi-habitats various, you need to choose the samples in each habitat in the distance of 100 meters long, and the number of samples should be selected like proportion habitat which occurs in this segment of stream. So, and also is recommendation that in segment of stream you can choose and collect, for example, no less than 10 samples, and if possible 20 or 30 samples units you can take, because so number of samples will reflect the true situation in the stream, and also so number of samples will be good for appropriate for statistical calculation. If you have a stream with a uniform bottom, like in this picture, you can choose a segment, shortest segment, and instead 100 meters long, you can choose only 10 meters long and take the samples in shorter distance. So sampling methods of invertebrates and devices that you need to choose for sampling depend on your project, on your research goal, on your task, depend on the stream or river condition, of course. And as we said, the invertebrates, bottom invertebrates are very diverse, so they all live on the bottom, but some of them live on the surface of bottom. Others hide under the stones. Some other groups live or burrows in the sediments, and therefore we are not sure that we collect all these invertebrates during one collection. So, of course, we need to choose suitable techniques, suitable equipment for something, but of course, we have no possibilities to collect majority of them using one technique or one devices. So we need to choose between them. And as I said, besides simple sampling of invertebrates, we need at the same time to collect and abiotic factors, which can reflect the physical state or condition of the water. And here I will present only some most common or simple methods for collection of water invertebrates. So, kicknet. Kicknet, more possible use like qualitative technique, because using this sampling we can collect composition or diverse various invertebrates. Sometimes we can use this method for quantitative evaluation, but for quantitative evaluation we need to choose some special area or determine the special time for sampling of invertebrates. And for kicknet sampling usually is used kicknet, which is quite large, about 1 square meter area, and have 2 handles. And one person keep the net faced up to stream, and another person using the foot or hand disturb the stones, and so try to dislodge the organism or invertebrates which go in the net, in the mouth of net by current. So kicknet, and also you can use D-frame deep net, which is possibly more useful because only one person can collect the samples. So we're using the same method. Because for this sampling need participate 2 persons. And so you simply can use the sticking or perform the sticking by foot, or as I said, by washing the stones, for example, using the hands. And this method is quite common for sampling invertebrates in the flowing water. And another method, server or hair samplers is used for this method, more qualitative. More qualitative method, because here we collect invertebrates from known area. And server sampler have 2 frames, and then this horizontal frame is placed on the bottom, and using hands with disturb or foot, we disturb the substrate and dislodge various invertebrates from known area. And therefore, this is not used for quantitative research. And the advanced version of server sampler is hair sampler. Hair sampler from metallic cylinder, and they also have a net. And this metallic cylinder have area similar like Cerber sampler, but provide or protect from escaping of invertebrates. Because you put this cylinder on the bottom, you push the hand inside, and you disturb the stones from this area. And so high metallic cylinder prevent or escaping of various invertebrates, because here, by using this sampler, some invertebrates can escape both sides of sampler. Next simple method, collect some substrate, stony substrate stones from the bottom of flowing water. One variant, what you simple can collect few stones if you have, for example, you investigate shallow and narrow stream, which have a cobble bottom. So you collect a few stones, one stone from, for example, from one edge, another stone from another edge, and a few stones from the middle of stream. And you bring the stones on the shores, and then you simply can scrap by using knife or some teeth brush. You can scrap invertebrates. And according to these invertebrates, you also can to say something about quality of water. But if you simply collect the stones, difficult is to provide this, perform this investigation for quantitative. Like quantitative research, because quite difficult is to calculate the shape of stone. You know that the stones have various shapes, and therefore to count the shape or area of a stone is difficult. And if you want to perform exactly or this quantitative research by using stones, you simply can use empty color slide film, only frame of film, and transparent film removed, and using this area, already known area, you can scrap the invertebrates from the stones only from so small area, and in this case you can obtain quite exactly known results from some special area. And also you can use artificial substrates, which is quite interesting and popular now. Like artificial substrates, you can use various things. You can use special wooden plates separated by each other with gaps, which allow enters for various invertebrates and colonize on these plates. Also, you can use an organic material like dried leaves, and dried leaf packet in the some sacks, for example, in the nets, and submerge this packet under water. You simply can attach to this leaf pack the bricks, which act like anchor, and then keep your sample artificial substrate under the water beside, more closer to bottom. But also at the same effort you can apply for collecting of invertebrates which live on the surface, for example. And then you can left this packet floating on the surface of water. So these methods, when nets filled by dried leaves, usually 15 or 30 grams of leaf used for sample. You can submerge underwater and left this artificial substrate for a week or 2 weeks, and after 1 or 2 weeks you check which invertebrates start to live on your artificial substrates, on your substrates. So quite suitable methods, because you will not need to spend time for collecting of samples. You simply submerge your sample, and you will check the sample at the same time, and then you will have some invertebrates which colonize your substrate. So, for example, here you can take a look how look the same artificial substrate, so wooden plates, grow it with which organism you can see here. Yeah, JSON polymorpha. So bivalve mollusk, and after a few weeks, so this not after a few weeks, after 1 month, you can see sow situation possible. Of course, it is dependent on the foreign water conditions. And here, for sampling and sorting of invertebrates in the lab possible, which you already have for samples taken from the streams and rivers. Of course, you can use quite simple equipment devices. You need to have a few white trays. You need to have some buckets, small dishes, sorting plates, and then some pipettes and tweezers or forceps for collecting. And in the lab, this collected material you sort in the vials or in some bottles and fix them with ethanol. And if you have very dense sample and numerous various organisms, you need to make subsamplers or divide the sample into subsamples. For subsamples, you can use some special like subsampler, which have just simple box, which have about 100 circular cells. And then when you fill the sample in this box, so you can take only 10 content from 10 cells. And these 10 cells can represent the whole sample. And if you have not some special sampler, you can use white tray graded into the quadrants. And if you have a high number of invertebrates, you can take only few quadrants from this grid, and these quadrants will represent your sample. So samplers and sample division into the samplers will do your work more rapidly and more easy. After fixation and identification of invertebrates, you need to label the fixed material for storage, and then in the labels you need to write collector's name, data, some abiotic factors, and of course site and condition where you collect the material. And identification, as I said, some groups require to identify them under some special species, but the majority of them you can identify under the level of families or orders. Here you can see the last tip. So metrics for benthic macroinvertebrates. These metrics or parameters of benthic macroinvertebrates, which can use like diagnostic tool. And then for like example of cell parameters, which are important and which you need to count and record. So it is a number of species. It is a number of collected individuals. It is number some sensitive invertebrates, sensitive to various stressors. And these metric parameters you can use for calculation various index, which use for evaluation of water quality. And of course exist various index. Here some few examples of benthic macroinvertebrates metrics like composition, richness, so species diversity, tolerance to various stress, and depend on these metrics, so they can see that how they provide the response for various stressors. Of course, if you have stream with diverse invertebrate and high number of various invertebrates after some perturbation and stressors, we can check and we can find that richness or diversity of invertebrates start to decrease. And of course, it will be quite important and significant parameters for measure and for determining water quality. And here you can see a few examples of indexes which can use for evaluation of water quality by using invertebrates and by using these metric parameters of invertebrates. So species diversity, taxa richness, APT index, which is based on count on families of invertebrates which are very sensitive for organisms. And some of these index, how calculate these indexes, you can find in the internet, simple online, and here few links, few address which you can use. You will find only formulas, and then you can enter your collected number of your invertebrates, and you can obtain results of calculation. So biotic index, which are based on abundance of organisms which tolerate stress or no. So very common index for evaluation of water quality. And for calculation, biotic index is important to know tolerance value of invertebrates. And according to this tolerance value, invertebrates are grouped into 3 groups. Some means of tolerance value can vary, slightly vary. It depends on countries and then depend on research, but if you will choose to calculate biotic index, and you need to know tolerance value of organisms, you need to check some research papers, research articles, and you will find in these articles tolerance value of some important invertebrates, according to which you can calculate biotic index. And here, like example of tolerance value, so points or scores from in one source you can find from 0, in another from 1. From 1 to 4 usually correspond. Sorry. Correspond more sensitive organisms which need to have quite good condition of water, for example, high number of high concentration of oxygen and so on. And on the end, opposite end of this arrow, we can find organisms which are evaluated by 8, 9, or 10. And usually these organisms correspond or represent the poor water conditions, low concentration of oxygen and high concentration of organic nutrient in the water. And after calculation biotic index, you can get some essential information about quality of your water. So here, like example, tolerance value of some invertebrates based on the families, not the species, but the families. And here the same sensitive group of organisms or invertebrates grouped according to the sensitivity reactions to the various stressors. So first group, sensitive to pollution, and you can see some organisms which represent this sensitive organism, which already have, not already, usually have sensitivity scores from 1 to 4. And the second group, largest group of invertebrates which can live in the clean water and in the water with moderate pollution. largest group. And the last group, it is organism or invertebrates which can survive in the water which contains small concentration or less concentration of oxygen, high concentration of organic nutrients, and they represent groups of invertebrates which can tolerate the stress. Of course, if you will find in the water this group and some numbers of first group, it is not mean that your stream is polluted. If you will find this organism from this second group together with the first group, but if you find only majority specimens from the second group, so you can suspect that your water moderate pollution. So it's quite useful because the majority of them wake up and now start to observe what happens. If we have short break, I would like to ask you. I said that invertebrates are divided into the groups according to sensitivity. Possible you already know which invertebrates represent the first group, organisms which are sensitive for pollution, sorry. Possible you know, and you can list the organisms which represent the first group, sensitive organisms for water pollution. Do you know some invertebrates? Can you list? Yeah.
Mayflies?
Ephemeroptera, mayflies. Yes, mayflies. Very nice. Larvae of mayflies, we can divide. They represent the first group, and but here is one note that not all larvae of Ephemeroptera represent the sensitive group of organisms. Not all larvae choose only good conditions. And when we need to evaluate water quality, depend, for example, on Ephemeroptera or mayflies larvae, sometimes we need to check the genus or species level, because some species of mayflies larvae we can find in the water which have moderate conditions. So, okay, Ephemeroptera or mayflies larvae. Crayfish. Crayfishes represent the second group. Crayfishes represent the second group, and they enter in the second group, which can live in the clean water and also can tolerate and moderate polluted water. So, the same second group, which do you know some of you how to explain odonata? Okay. Yes, dragonfly. Do you know dragonfly? Okay, so dragonfly, the larvae live in the water, and then usually some of them represent can enter in the second group, and some in the third group, but majority of them enters in the second group. So here is some additional for the first group, or the sensitive organisms, stoneflies or larvae of stoneflies. So stoneflies, how look the adults? Quite common, and you can recognize them according to these two tails on the end of a body. And here the larvae, and larvae, stoneflies larvae, yes, we can say it is true indicators, good indicators of good water quality, because majority of them need to high concentration of oxygen. They like strong velocity. They like great supply of oxygen. They like stony bottom. So therefore, these larvae they try to live only in soil conditions, and therefore they are good indicators of water quality. And the larvae we can recognize according to these two tails, which have an adults too. But and the second features of these larvas that we avoid these details. So two enough clearly visible claw on end of each legs, and also gills, which this larva for respiration, tracheal gills, they locate on the basis of legs. So in the thorax region, you can find the gills. And according to these few features, you can identify these larvae quite easy. I talk about order, so you can identify that there are stoneflies. And now it will be okay, because majority of them are good indicators of good water. And as you said already, so larvae of Ephemeroptera, mayflies larvae also depend enters into these 5 groups, sensitive organisms, which have tolerance index from 1 to 4. And how look mayflies adult, and how look the larvae from a first look, possible they are similar to larvae of stoneflies, but Ephemeroptera larvae usually have 3 tails. Sometimes some species have 2, but the majority of them have 3 tails. And another different feature that these larvae have tracheal gills, which locates not in the thorax region, like in the case of stoneflies, but in the abdomen region. So these tracheal gills, or plates for respiration, locates on the abdomen. And according to this feature, you can quite easily identify larvae of mayflies. As I said previously, mayflies live very short. Like adults, they live very short in the terrestrial environment. After finishing of their metamorphosis, adults fly, extend their wings, and they simply try to find the partner for population, try to populate, produce the eggs, lay the eggs, and simply die. So their life in the terrestrial environment is very short, and sometimes can last only 1 day, or some species only a few hours. So they fly from the water, reproduce, and die. Meanwhile, vulva life spent in the water condition like larvae or larva stage. And exist various larvae which various of larvae and species which feeds on various organic particles, plant material, shredders, scrubbers, and so on. But in the case of mayflies larvae, more or less we need to identify not only order, but look more deeply and the level of families possible, or genus. And here the third group, Caddisfly Trichoptera. Adults look like moth or butterfly, not so colorful, and then have their wings covered by hair, and larvae. Here I wrote the exactly names of families, because majority of these larvae, they represent the second group. They enter in the second group, which can live in the moderate polluted water, majority of caddisfly larvae. And some of them, only some of them, choose to live in the water which have high content of oxygen, and then they choose to live in the clean water. So some special family. And only this specimen from this family enters into the intersensitive group. And the characteristic feature of caddisfly larvae that we can make the case or hole which surround the body, and according to the shape and content of composition of a case, we can quite easily identify these organisms, different from adults, because for identification of adults, you need to have more knowledge than of larvae. Okay. And here's large group, second group of organisms, various invertebrates, which live in moderate polluted water, or also at the same time they can live in the clean water. So majority of various invertebrates represent or enters into this group. And here I will present only a few of them. So you can see Odonata, so larvae of dragonfly and then damselfly enters here. Crayfish also, various crayfish also here in this group. As I said, majority of caddisfly larvae enters or depend also for this group. And cranefly larvae. Cranefly, do you know cranefly? Diptera. Diptera like big mosquito, which have long legs and 2 wings. So some related to mosquito. And here the clams, mussels and clams are the bivalve mollusks, which also enters into this group in the organism which can live in various waters. And here how look the larvae of Odonata and dragonfly. Possible you know how distinguish between dragonfly and damselfly. Here's damselfly. Damselfly are smaller and can fold the wings together. Yes, damselfly. And damselfly larvae look so. They have long and narrow body and have some special leaf or gills which look like leaf on the end of body, on the end of abdomen. So larvae of damselfly. And dragonfly bigger, and they have wings constantly extended. They have not capability to fold these wings on the back. They constantly keep these wings extended. And how look the larvae of dragonfly? They have enough short, wide, strong body, big head, big eyes, and they have not the gills on the abdomen. Possible you know how they breathe. So these use for respiration these tracheal gills, and these larvae, they respirate quite interestingly. They involve the water together with oxygen through the anus in the hind intestine. And then beside the hind intestine locates trachea, and then they obtain oxygen from water in this hind intestine. And then after they push water out through the anus. So quite interesting method for respiration, and some similar method for respiration also you can observe in some water turtles, some from reptiles. They also can respirate through the anus. Okay, and here is the very characteristic feature of all larvae of dragonflies and damselflies. All larvae, dragonflies and damselflies are predators. And how fits the adults? What eat the adults?
They're also predators.
Yeah, they're also predators, and larvae and adults are predators, and they eat various other insects. Despite the larva type, all larvae of dragonflies and damselflies have some special labium or mask, which locates below the head. And this mask or this labium can be extended very rapidly in some distance, and this mask on the tip have also spines or like forceps, And this mask is used for capturing of prey through some distance. And this mask helps for larva capturing the prey. So according to the mask, you can identify that larva belong to the order Odonata, according to the mask. And so here how look the mask in the rest, and here how look the mask extended. So larva can very rapidly extend this mask and capture the invertebrate. So another additional organism from the order Megaloptera, which represent also this second group, and how look the larva of Megaloptera have, because predators have clearly visible mandibles, some special lateral filaments, and tail which look like spine. Okay, Trichoptera, we already saw. So various species of Trichoptera or caddisfly larvae live in the water and build the various cases around which surround the body. Of course, exist some larvae of Trichoptera which have not produced the houses, but they simply make the nets, and then using these nets collect material from the water. And here some example of Coleoptera, Elmidae family beetles you can find in the water also. And what is different from majority of beetles, in the water you can find an larva and adults. Because usually you can find only larvae, but in this case you can find an adult, and in some source, in some papers, you can find information that this Elmidae family beetles, they represent good conditions and represent or indicate good supply of oxygen. But in another source, you can find that these beetles belong to the second group and represent moderate pollutant group. So it is dependent on source. And so here is this, as I said, mosquito which have long legs and belong to the order Diptera. So simple fly mosquitoes belong to the same order, and some larvae mosquito also can develop in moderate polluted water. A characteristic feature of majority larvae of Diptera, which allow to them say that this larva belong to the order Diptera. It is that they have not the clearly visible legs, and majority of them have not clearly visible head. Because here, for example, in this picture, this end is posterior end, and the head locates here. So usually these larvae of various Diptera, they have not clearly visible here head and have not the legs. Of course, exist some exceptions, but majority of them have so characteristics. And bivalvia mollusks, which have two shells, especially characteristic like indicators. And here you see the features which make them very advantage and useful like indicators. Because we more or less that they can move, of course, but more time they spend in the same place. They come enough long life of them, so they can live several years at the same place, and they filtrate the water, so they constantly live at the same place in the same water and enough long. So it means that they can accumulate, concentrate various material which are in the water. They also can accumulate heavy metals, and therefore there are very suitable indicators for the water quality. And the last group, so organisms which like pollution in the water, and they compete with other organisms, and if in the stream or river you cannot find any other organism and only specimens which represent this group, of course you can suspect that your water is polluted. And here is some common indicators from this group. Larvae or Diptera larvae from a family Chironomidae. by another word, bloodworms. Usually they contains a high concentration of hemoglobin in the body, which allow to them live under conditions of low oxygen. Here characteristic feature of these larvae, they have curved body, and they have some not true legs, like prolapse, on the end of the body and beside the head. And according to these prolapse, you can distinguish which larvae. And also larvae from the family Diptera, which are filtrators, and characteristic feature that they have special filter fans on the head, which helps to collect various materials from the water, and have some bulky or sweep posterior end of the body. And aquatic oligothads, aquatic worms, if you find on the bottom quite large number, so oligothads like tubifex, which have segmented body, so some organisms related with earthworm, and also quite large number, some special species of earthworms which can live in the water, and leeches. You can suspect that your water is not a very good condition and have low oxygen concentration, and possible contains or have high organic nutrients. And gastropods mollusks. We said that bivalve mollusks, they enters into the second group. Meanwhile, gastropods mollusks enters into the third group, which tolerate pollution. Possible you know why, or you can explain why. Why bivalve mollusks, clams, mussels?
They can also live in a terrestrial habitat.
These species no, but we can say that, for example, these and these Planorbis and these Bithynia can say that there are amphibious. Yes, but you are very close to the right answer, because for respiration, these mollusks use some special mantle lung, and they for respiration they use for they use oxygen from air. So they can rise from the water, polluted water, then can rise to the surface, and they simple breathe using air, and therefore they can live enough in polluted water environment. And here are some research, some source in which you can find various keys for identifications of various macroinvertebrates.